Did you know there are species of fish who are able to survive in conditions that other animals wouldn't? And did you know that the research being done to study these species of fish is being done right here at K-State? Meet Dr. Mickey Tobler and his team of researchers.
My lab primarily studies animals in extreme environments. Extreme environments are environments that are characterized by harsh environmental conditions that most organisms can tolerate, and we're really trying to figure out what kind of animals have managed to colonize these types of environments and how do they actually manage to survive while most other organisms don't. We're trying to do this, the way we're trying to understand life in extreme environment is really to study these small life-bearing fishes that have managed to colonize springs and streams that are rich in toxic hydrogen sulfide.
These fish are known as Placida mexicana, or more commonly known as the Atlantic molly. This species of fish are found in sulfur springs throughout Latin America, where Dr. Tobler's team catches them and brings them back to their lab to study along with Kansas State University graduate students.
We go out to the field in Mexico where most of these sulfide streams are, and we bring the fish back here where we're going to test them for swimming performance as well as breathing performance, as well as measure their different body types.
So what we do is we go in the field, we take fish tissues from these different populations, and then we sequence the DNA in that tissue, and we get back a bunch of data that we have to analyze on the computer. So a lot of my work is based around writing code and things like that on the computer.
First sight, some of these sulfur springs look very similar to other streams you might find, but they're really characterized by having this baby blue coloration. That coloration comes from sulfur compounds that gives the water this distinct coloration. Most animals, when they're exposed, they basically fail to be able to produce energy and they will die. But somehow these fish have managed to has that problem. And so we're really trying to understand how the fish manage to detoxify or tolerate the hydrogen sulfide in those springs.
Previous research has been focusing on genetic aspects of research. So they look at what genes have been affected by living in hydrogen sulfide, and other research is focused on what the fish do in the environment. But we have no data that connects the two of those together. So what I'm doing is actually measuring functional traits so that we can relate how these genetic changes actually matter in the face of environments.
There is research being done here at K-State about this subject. Dr. Tobler and his team are not alone in their endeavors.
A lot of people actually involved in this project. It's not only scientists here from Kansas State University, but we're also collaborating with scientists in Oklahoma at Oklahoma State University in Stillwater and at Washington State University in Pullman, Washington.
The majority of the work that I do is in the lab where we're sequencing genomes and sequencing transcriptomes, and then the bulk of the things that we do are on the computer. Each of us brings a unique perspective to the group so that we're able to answer a larger scientific question that none of us would be able to answer on our own. And for me, that's the really exciting part, is that we're able to come together and bring each of our unique expertise and the expertise of our trainees that are also working on it and answer a bigger question that certainly in my lab we wouldn't be able to do just on our own.
The specific goals of our current project is really to understand how the fish adapt to these extreme environments at different levels. We want to understand what genes are modified within the genome of the fish, what are the genetic differences between tolerant and non-tolerant populations.
Dr. Tobler and his team hope to provide a glimpse to the limitations of other organisms to extreme environments through these fish, and to see the biodiversity, especially with humans and climate change. What's this button do?